Hey guys, hi. Welcome to the next session of Unacademy. We are having back-to-back 3 sessions, and after that, after each YouTube session, we are having an open house session also on the Unacademy app. Open house is a new feature that is introduced by Unacademy that is specially dedicated for one-to-one audio interaction with the learner and the educator. Okay, so all those who have not yet understood about open house, I'm going to brief you guys about that in this session only. And right now, this session is for understanding about translation. Okay, I got it. So I'm your educator for biochemistry. I am Dr. Shivangi Shukla. I teach over two platforms, that is Unacademy Future Doctors, here the Unacademy channel on YouTube, as well as on the Unacademy app. Okay, and I have been taking a lot of classes covering the entire syllabus of biochemistry. I have taken revision sessions, I've taken MCQ sessions. Today at 10:45, I have an MCQ bonus biochemistry session, that is MBBS session on the Unacademy app. So stay tuned to that and do join me in that session as well. Okay, I'll just quickly brief you about the different types of subscriptions available for NEET PG on the Unacademy app, the Plus as well as the Iconic subscription. Both of these will give you access to video resources from top educators across the country. Video resources both live as well as recorded, which can be accessed from any device of your choice. Okay, be it your phone, your laptop, your tablet, your PC, whatever. And even while on the go, you can have ready access to these. Okay, the Iconic subscription will also give you access to resources from Prep Ladder. Okay, so it is also called Best from Two of the Best, isn't it? Now, to avail additional 10% discount over and above the already subsidized prices, do not forget to put in the educator code DRSHUKLA, Dr. Shukla. This code is going to fetch you an additional 10% discount over and above the already subsidized prices of the in the app. Okay, Dr. Shukla, DRSHUKLA, Dr. Shukla. All right. So let us start with translation. What is translation? If anyone wants to know the definition, see, we have just finished with an open house session regarding the strategy for answer writing. Okay, now we are starting with the taking back from that, if you have to write a short note on translation, then what did we discuss in the strategy session? We discussed that the most important part is the structure of your answer. Your answer should be properly structured. So in any short note, first of all, you should have an introduction, a short introduction, where the best part in any introduction is to define. Okay, the best approach is to define something. So define translation, okay, in your introduction. What is translation? Translation is the process of um, formation of proteins, formation of amino acids from the information contained in the mRNA. And that information in the mRNA has been transcribed from the DNA. Okay, understood? So translation is a process that involves the manifestation of the genetic code into amino acids. Okay, now what happens is here, so all in all, what happens, the mRNA molecules, whatever information they have, that is translated into amino acids. But mRNA and amino acids, both of them don't have any affinity for each other. They cannot speak directly to each other like we can speak during our open house sessions. Okay, so they cannot speak directly to each other. So what do? They need a mediator. They need an adapter molecule indirectly, via which they can communicate with each other. Okay, so what happens is these mRNA molecules will bind, they will be read by tRNA. Okay, and that tRNA will then synthesize proteins by attachment of different amino acids together in a sequence. Okay, please pay attention to all the keywords that I utter. Okay, the keywords are very important for writing any answer. Okay, now there are a few important terminologies that you should know for your before you start with the basics of translation. The first is triplet codon. Now, can any of you tell me what is the meaning of triplet codon? What is a triplet codon? Why is there a need for a triple letter code? Triplet means a triple letter code. Why is there need for a triple letter genetic code? Can any of you tell me why? See, what happens is there are 20 amino acids. All of you know this. And as we saw that the amino acids are translated from the genes. Okay, the genes or the genes that are present in the DNA. So, DNA contains how many nitrogenous bases? We just discussed in the introduction part, the first session tonight in the evening. DNA contains 4 types of nitrogenous bases, A, T, C, G. I'm not considering U because U is synonymous with T only. Okay, so there are 4 types of nitrogenous bases. Now, and they have to code for 20 amino acids in different permutations and combinations. Now, if suppose we have a code containing just one nitrogen base, be there 4 to the power 1, that means there will be 4 such codes. Now, there will be just 4 codes and we have 20 amino acids. So, what will happen about the 16 residual acids? They also want some codes. Understood? There will be no codes for them. So, what will happen? Now, consider 2 nitrogenous bases. 4 to the power 2. So, this will be 16. So, if each code contains 2 nitrogenous bases, then they can code for maximum 16 amino acids. Still, we have 4 residual amino acids with no code of their own. Okay. So, next consider 3 nitrogenous bases. 4 to the power 3 is what? All the maths wizards, it is 64. So, now we have cracked the code, isn't it? 64. If we take 3 nitrogenous bases to code for each amino acid in different permutations and combinations, then we'll come up with 64 codes. And these 64 codes are sufficient to code for 20 amino acids, isn't it? So, you can say that 64 codes will code for 64, there are 64 codes for coding for 20 amino acids. Now, what will happen? Codes are extra and amino acids are few. So, more than one code can be there for one amino acid, isn't it? More than one code can be there for one amino acid, and that is what brings us to the first characteristic of the genetic code, that is degeneracy. What is degeneracy of the code? That is more than one codon can code for one amino acid. More than one codon can code for one amino acid. This is the degeneracy of the genetic code. Now, for those of you who are observant, you should say, here it is written 64. We have come up with 64. But here you are saying, you have written just 61. Where are the 3 codons? Where have they gone? These 3 codons, my dear children, they are UAG, UAA and UGA. And why are they not coding for any amino acid? Because they are stop codons. Before I started with this session, in the last session I mentioned, what are the 3 steps of translation? Initiation, elongation and termination. So, how does translation terminate? Whenever any stop codon is encountered, then translation is terminated. Okay? Are you guys following? Now these stop codons, what are these? UAG. UAG is called amber. How will you remember this? This is AG, anger, anger. So it is resembling amber. Next is UAA and UGA. Okay, now UGA can be called Gopal. Gopal. So this is opal. And the last one remaining is ochre. Got it? So these are the names for these. You should know because they'll be asked in your PG entrance examinations. Okay. UAG, amber, UAA, ochre, UGA, opal. These three are stop codons. That is why it leaves us with 61 codons coding for 20 amino acids. So therefore, one amino acid can have more than one codon. Okay, but this leads us to the second characteristic, which is one codon is available for only one amino acid. Now you understood? One amino acid can have more than one codons, but the opposite is not true. Okay, one amino acid can have more than one codons, but one particular codon will code for only one amino acid. Okay, so one code is unambiguous. If you see UAG, you know that this is stop codon only. This cannot be anything else. But all these three can behave as stop codons, but they cannot behave as anything else. Understood? So there is degeneracy of stop codons as well. And also there is unambiguity of stop codons. Understood? Next is non-overlap. Okay, the reading of codons on the mRNA is non-overlapping. So suppose this is the mRNA molecule. It has AAG, CUU, 2AA. So it cannot be read as, and this is something like this, okay? Cannot be read as this and then this. No, it cannot overlap like this. Whatever it is, it has to be read in a similar fashion. It cannot overlap. Okay, so that the code is read similarly. Okay. Similar characteristic is it is non-punctuated. That means there should not be any gap that should be present in between. It is a continuous sequence until you encounter a stop codon. Non-punctuated, non-overlapping, unambiguous, degeneracy. And the fifth one is universal. That is, in every individual, this particular codon will code for this amino acid only. But there is some exception to it between the cytoplasm and the mitochondrial DNA. Now, the nuclear DNA is translated in the cytoplasm. Okay, but the mitochondrial DNA, mitochondria also has DNA, if you would know. Okay, the mitochondrial DNA can, the same codon in the mitochondrial DNA can mean something else, and it will mean something totally different in the cytoplasm. Okay, so AUA in the cytoplasm is isoleucine, which is an essential amino acid. AUA in mitochondria is methionine, which is also an essential amino acid. Now, AGA and AGG are both arginine. You remember this is degeneracy. That means arginine can be coded for by more than one amino acid. But AGA and AGG are stop codons for mitochondria. Stop codons in mitochondria are AGA, AGG. Stop codons in cytoplasm are UAA, UGA, UAG. Okay? Understood this? This is the exception to universality. In all the individuals, AGA, AGG is going to be arginine in cytoplasm in all humans. But in the mitochondria, in all humans, it will be stop codon. Understood? This is an exception to universality. Alright guys, if there's any doubt, you can tell me. Okay. Now a brief about splicing. Now after transcription, what has been formed is the primary RNA transcript. Now it has to undergo a lot of modifications, like capping, tailing, like methyl guanosine cap will be added. The methyl group in this is coming from S-adenosyl methionine. Remember S-adenosyl methionine? Where have we heard of it? Come on, come on. Tell me in the comment section. Where have we heard of S-adenosyl methionine? S-adenosyl methionine is a universal donor, methyl donor in our body. Where is it coming from? The methionine amino acid, it gets converted to S-adenosyl methionine, which then gets converted to S-adenosyl homocysteine and then forms homocysteine. This is conversion of methionine to homocysteine, which is cyclical. Homocysteine will again go back to methionine utilizing vitamin B12, and this involves one carbon transfer of methyl group between tetrahydrofolate, B12 and homocysteine and methionine. Remember that? Remember, all of you, very important. So, see, from one topic, I have jumped to the other topic. This is how you should revise, integrating different topics together. Very important. Okay, understood? Always integrate different topics together. Anyway, so, yeah. So what is splicing? So capping was this. Tailing is what? Addition of poly A tail, polyadenine tail at the 3' end. Capping is at the 5' end. Following all of you, if there's any doubt, do let me know. Let me just check the comments if there's any doubt, because I have been teaching for quite a lot of time. Let us pause. Let us give a break for 1 minute to see if there's any doubt. Any one of you can post your doubts, okay? Okay, so in modifications, what did we see? There were 3 modifications. One was there are 3 modifications. The first one is the capping. Second is tailing, and third is splicing. Splicing is also very, very important. Okay, what is splicing? Let us see. Splicing is when the primary RNA transcript that has been formed fresh hot bun. Okay, primary RNA transcript is acted upon by small nuclear RNAs and other proteins to form a spliceosome. Okay, now this RNA, excuse me, now this RNA transcript, it contains some extra portions. Okay, what are these extra portions? These extra portions are called introns, intervening segments. Okay, normally this RNA, it has the genetic material, okay, that has been transcribed from the DNA. Okay, now this genetic material, it is present in the exons. Okay, but it has some intervening segments which have genes, but they are not to be translated into proteins, and these intervening segments have junk DNA, and they are called introns, intervening segments. They have to be removed, they have to be trimmed out from the RNA. Okay, now how is that done? By the formation of a spliceosome. Spliceosome is what? Spliceosome is a body. Zome means body. Spliceosome means that helps in splicing. So this spliceosome is formed when the primary RNA transcript combines with small nuclear RNAs. Remember, in our last session, what did we read? There were 4 classes of RNAs. Okay, tRNA, mRNA, rRNA, and small RNAs. small RNA contains two types, that is small nuclear RNA and microRNA. Remember? So all these, what did I tell you? In editing and processing, these were involved. So this is splicing is a type of processing only, processing of the primary RNA transcript. So here what are, now can you see one application of small nuclear RNAs? U1, U2, U4, U5 and U6. Apart from U3, all of them. So U1, U2, U4, U5 and U6 are the small nuclear RNAs that will combine with the primary RNA transcript along with other proteins to form the spliceosome. Understood? So, I already told you about the exon and the intron. Isn't it? Introns are what? Introns are intervening segments. And what are exons? Exons are the ones that have to be translated. Okay. Now, this is the primary transcript. Fresh, hot RNA buns. Okay. Now, you have to remove the burnt part. The burnt part that is useless from this bun. Burnt part is the intron. How will it be removed? You have to apply some tongs. Okay. Now these tongs are the spliceosomes. Okay. There will be a nucleophilic attack at the 5' end. See, this is the 5' end. At the 5' end of the intron, and it will curl upon itself to form a latch or a lariat. Lariat is a latch, a hook. Okay. So it is like a tong. Now this will be removed from here also, from the 3' end also, and both the exon terminals will then be attached together, ligated together to form the modified RNA. Okay, this is the mRNA which will now undergo translation. Understood? Following? Excuse me. If there's any doubt, you can tell me. Hi, Jatin. Jatin is saying, ma'am, why are we not able to do reverse translation? Can you tell me one utility of reverse translation? If you have asked me the question, I'll help you develop your thought process. Tell me one utility of reverse translation. Tell me. One utility of going from amino acids to DNA. Then back to DNA through reverse transcript. Tell me. Come on, I'm waiting. Tell me one utility of going from amino acids to genes. Tell me. Let us develop, let us give wings to your thoughts. Okay. What is the utility of going from amino acids towards genes? Tell me. Tell me, come on. No? Those amino acids go back and synthesize the correct genes that we can infuse. So we can put in those amino acids along with the requisite tRNA or whatever adapter molecule and the enzymes, and they'll be able to synthesize the correct genes. So genetic diseases can be treated. Okay. Genetic diseases can be treated using that. Alright. Okay, so let us start with tRNA. Okay. Now, before that, let me just remind you, at 8:30, that is just going to have the session, the open house session, another open house session at 8:30, and that is on biomolecules, metabolism of biomolecules. That will be open to your doubts. Okay. I'll first of all, I'll wait for you to put up your doubts, and then I'll summarize most the metabolism part. And also I'll tell you the important topics, the high yield topics from the metabolism chapter that you have to study. So that is the basic purpose. Also, you can please put up all your doubts and attend that open house session. Okay, attend the session, the open house session, and that will be live at 8:30 on the Unacademy app. You have to open the Unacademy Learner app, go to the NEET PG section, and look up for Shivangi Shukla. There you will find the button. You will be taken to the live chat room. There we can have a 1:1 live audio chat where you can ask me your doubts regarding the preparation, regarding, because the session is specially dedicated to biomolecules. So any doubts that you have regarding biomolecules, you can attend that. Jatin, that is specially for plus and iconic subscribers. I think that is why you're not able to see. Okay, because that is specially for, currently it is for plus and iconic subscribers. Okay, the open house session. So a lot of people are benefiting from that. They are asking their doubts, clearing their doubts regarding preparation at any, because I am taking 3 open house sessions per day. So you can you can ask any of your doubts regarding preparation, whatever doubts, any MCQs that you're not able to solve, any answer that you feel that you have not got the requisite amount of marks. So you can ask me why, what is lagging in this particular answer. So all these things we can discuss. Okay, also the strategy sessions, reading sessions, how you should spend, how much of your time you should spend reading, how you should try and read, how you should design your study pattern. All these things are what we are going to discuss there. Okay, so let us briefly start with the introduction of tRNA. Now tRNA, as I already told you, is an adapter molecule. So what does this adapter molecule do? It connects mRNA with amino acid. Why? Because mRNA and amino acids don't have any affinity for each other. So tRNA, being the adapter molecule, will connect them. Okay. Now, as an adapter molecule, the first step is charging of tRNA with the specific amino acid. Just a few days back, while discussing MCQs, we discussed the step of charging of tRNA molecule with the specific amino acid. What is charging? When the tRNA molecule is associated with a particular amino acid for which it is carrying the anticodon. Okay, charge the tRNA, the specific tRNA. Understood? So there's a lot of specificity. And what is this enzyme that is going to charge? It is amino acyl tRNA synthetase. Synthetase. Why am I singing this? Singing along here. Why? Because I want you to focus on synthetase. Synthetase, that means requires ATP. And where is this ATP? Can you see adenosine? Understood. Amino acyl tRNA synthetase enzyme is going to form a complex with adenosine from ATP, and in that complex it will bind the tRNA. So what will this tRNA become? This tRNA will have, finally this complex will have transfer the amino acid to the tRNA, and then the enzyme complex will be replenished. Understood? So this is what, how is this enzyme behaving? Like covalent modification, ping pong mechanism. Go back to the chapter on enzymes and then you'll remember, then you'll understand. Okay, understood. I think now the first step we have completed. First step is the charging of tRNA molecules. Understood? The first step is the charging of tRNA molecules by the specific enzyme with the specific amino acid. Okay, this is the first step. All right. Understood. Now we have to read the structure of tRNA, which is very important, but this we'll read tomorrow. Okay, because currently I think I'm going live on the Unacademy platform on the open house session. All of you here, kindly join me on the open house session. Okay, I'm live there currently. Please come up with your doubts regarding biomolecules. We'll be discussing on the high yield topics, most important topics to be studied in different biomolecules that are there: carbohydrates, lipids, proteins, and we have discussed quite a lot about biomolecules. So we'll be taking it taking it from there on the app. So please join me there on the open house, in the open house session. I am live right now on the app, Unacademy app. Any doubts regarding your preparation, you can come and ask there. Okay, every day 3 such sessions. After each YouTube session, I'm taking 3 such sessions on the app. Okay. All right, if there's any doubt from this session also, you can take it there. From YouTube also, you can take it. Okay, I'll be available there. Okay, if there's any doubt, you can post it here. Otherwise, please come on everyone, hurry up. That is why I have not stopped the streaming right now, so that you guys don't face any difficulty in transitioning from here to the Unacademy app. Okay, so that you don't have any difficulty there. And please come on, join me there on the app, because the open house session has already started. That is why I have not ended the stream right now. Sale